Welcome, folks, to another AP Bio exam review session. I am so happy to have you here with me and to be right here with you. And what would make me really happy would be two things. The first thing is if you would just put in the comment thread a note telling me where you are coming from, where in the world you are coming from, and second, if my audio is okay and you can hear me. So I would just love to know about those two things and let me know. So I'm seeing that there are already a couple of people who are here, but I'm not seeing any feedback letting me know that people are hearing me and people can actually get what I'm saying, and that's an important thing. So if you've got some audio coming through, then let me know. And thank you so much. We got folks from Ohio. We got folks from Connecticut. Audio is good. Thank you very much, Ava. And Iraqi Kurdistan. And that is so exciting to me to be listened to from somebody on the other side of this planet. That is really fantastic. Ava, welcome. California, that's fantastic too. We got people from Vancouver, Canada, and that is really great. So I'm really happy to have you here. We started this process last night, though I've actually been doing some other lives even before that. And it's just great to be here and to help people learn what is, my favorite subject, which is biology. Super happy to be here. And got folks from Texas, Utah, Georgia. That is just thrilling to me. So what we're going to do today is two things. We are going to talk just a little bit about how to review for the AP Bio exam. I've already done that quite a bit, and I'm going to refer you to some resources to go into depth about that, because I know that there are people who have been here several times and they don't want to hear the same thing over and over again. So I'm going to refer you to some resources for that. I'm going to answer any questions that you have about Unit 3. If we're overwhelmed with questions, I'm going to encourage you to use Super Chat or Super Stickers. Just, help me out a little bit to get yourself to the top. And that's going to be a really fun experience for me if I see those Super Chats, Super Stickers come on in. And we'll do some practice multiple choice questions that I've been putting together. So that is the plan for tonight. I told you last night that we were going to talk about Unit 3. So I hope you brought your Unit 3 questions. And I'm going to change the view over here so just we can focus on what is important, and what is important is the information. So let's go like this, and no, that's not really where I want to be. That's where I want to be. Fantastic. So let's talk a little bit about how to review for the AP Bio exam before we get too deeply into this. I want to emphasize that everybody here who's watching me has a lot of time to review, but that's only true if you make a lot of time. So the first step to a 5, I didn't even put on the list, but you've got to work hard. There are going to be people who you're going to be competing with who are going to be working hard, putting in, hours a day, getting themselves ready for the AP Bio exam. You should be one of those people. AP Bio is hard. The ideas are deep. There are so many concepts to understand. And reviewing for a test like this is a big deal. So put in the time over the next 2 weeks. And that's really funny that someone just noted that they're used to listening to me on double speed. That's how I listen to YouTube too. Okay, so the first thing is I want you to use our 2-week plan, because what I've done is I've laid out a day-by-day plan, what you should be doing every day between now and the AP Bio exam. And if you set aside those couple of hours every single day, you're going to get there. You're going to get to a place where you walk into the test with confidence and you walk out knowing, I crushed this. I crushed this. And that is the experience that I want everybody to have. To get there, you've got to do a lot of what I call authentic practice. And that means you've got to practice in a way that mimics the task that you're going to be confronted with on May 5th. And that means I want you to write lots of FRQs. And you can do those from the College Board. I talked about this in my previous video. The College Board has been extraordinarily generous about posting past FRQs online. And they don't only post the FRQs, they post the scoring guides. So what is writing FRQs looking like? It means going to the College Board's website. You can just search for past AP Bio FRQs. You'll get everything from like 2024 to 2008. And just open up the FRQ, turn off your phone, eliminate distractions, and start writing. When you're done, look at the scoring guide and see how well you did. So that's one thing you can do. The other thing I want to encourage you to do is on my website, learn-biology.com. We have enhanced FRQs with personalized AI scoring. And if that sounds cool, it is. Basically, what you do is you'll see a practice FRQ. There'll be a little box where you can type in your answer, and then we'll give you feedback. We'll compare your answer to a sample answer. It's a fantastic learning experience. I've watched classes doing this, and it is really a magical experience, and it's going to make you improve so much. It's as if you had me there by your side telling you what was good about your FRQ and what needed to be strengthened. So give yourself that experience. And of course, to do that, you have to sign up for learn-biology.com in order to access that. And the way that you do that is you go to learn-biology.com/AP Biology and you sign up. It's not free, but it's not expensive either. And it's the best investment that you can make because it is designed for your AP Bio success. So don't wait on that because the sooner you do it, the more time you'll be able to spend with these fantastic resources. And then what I want you to do is answer lots of multiple choice questions. And I want you to do that in the same place on learn-biology.com, where we have something like 470 multiple choice questions that span every part of the curriculum. So if you're thinking, okay, I'm going to do this on my own, don't do it on your own. Don't do it on your own. There is support waiting for you at learn-biology.com. It is College Board approved. It is used as a textbook by hundreds of teachers around the United States and around the world. It's cognitive science based. We've done all the work for you of getting you ready. We've got multiple choice questions, enhanced FRQs, flashcards with personalized feedback that I was telling you about. And there's an app. It's called Biomania that gives you much of the content so you can study when you're on the go, when you're on the bus, when you're being driven somewhere, when not when you're driving somewhere, but when you're being driven somewhere. It's a fantastic resource. Don't wait. Go to learn-biology.com and sign yourself up. Okay. So Unit 3. All right. What are your questions? You got questions? We can do anything. We've got any of these topics. But before we do that, we have someone who has a somewhat interesting name. I'm just going to say Daily Dose. How do you not stress about the exam? What's the best way to study using the course and exam description? So the course and exam description is a fantastic resource, but it ain't the most friendly document that's ever been written. And in fact, what I would encourage you to do is use something that you can get from me. Just go to apbiosuccess.com/checklist. Ooh, misspelled that. Okay, so I just put that in the chat, apbiosuccess.com/checklist. And that's my list, which I based from the course and exam description that lists all the topics that you need to know in the friendliest of ways. So I don't think you should actually be in the CED right now. The CED is the course and exam description. It's fantastic, but don't use the CED. I would suggest, and of course I have a biased opinion here, learn-biology.com. We've done all the work of organizing the materials, and what that means is that the work that you have to do is just learning. So give yourself a break and do that. So again, just want to emphasize, if you want your question to get priority, use a super chat or a super sticker. Otherwise, I'm just going to go in the level of detail, and I'll start with Mohammed's question because Mohammed's probably furthest away, and I think it's also very late, Mohammed. You're amazing. Is it important to know every detail about photosynthesis and cellular respiration? No, you don't need to know every detail. You need to know the big picture. So why don't we just start with this? I think we're going to just go right to cellular respiration. Let's go right here. Okay. So cellular respiration is about making ATP. ATP, adenosine triphosphate. And like this diagram, you should know in the back, like the back of your hand, here's ATP. Cells can break off this third phosphate. They can hydrolyze this bond. And what that does is it makes energy available for the cell to do work. When you contract a muscle, when you think a thought, ATP is what's powering those processes. So after you take ATP, break off that bond, you make it into ATP and phosphate. Well, then you need to regenerate ATP, and that takes energy. There are basically two ways in which that happens. One is cellular respiration, and the other is photosynthesis. And those processes have a lot of similarities, and those are the two great metabolic processes that are running on Earth. And Josue is saying, is this energy coupling? Yes, this is actually energy coupling. And Daily Dose, if you didn't get the checklist, it should get emailed to you. And if it doesn't, then I'll show you how to contact me at the end of the show. All right. So you should know about the function of ATP. It's got a 5-carbon sugar. It's called ribose. It's got the nitrogenous base adenine, and it's got 3 phosphate groups. And the function is to power work within cells. Every cell makes its own ATP. There's no sharing of ATP between cells. If you've ever seen someone who was in the hospital and they needed to be on intravenous fluids, maybe they needed to be getting some energy that was going directly into their veins, they weren't giving them ATP. They were giving them glucose so that they could do cellular respiration. ATP is something that works at the cellular level. Here's the cycle. To store energy, cells take energy from food or light, and they use it to make ATP from ADP and phosphate. And to release energy from work, they do the complementary reaction. They remove a phosphate group from ATP, creating ADP and phosphate. Let's talk about the big picture about cellular respiration. And the big picture about cellular respiration is this. Somebody asked earlier, I think it was Mohammed, what detail do you have to know? This diagram, as scary and as intimidating as it is, this is about the level that you need to understand cellular respiration. You might need to go a little bit deeper than this. And looking at a question of how much ATP is produced from one glucose, I think it's between 30 and 32 is the answer, but you can look that kind of thing up. The main thing to know is where that ATP is created. And if you look at this diagram, you can see that there's a little bit of ATP that's created during glycolysis. There's a little bit more that's created during the Krebs cycle. And then most of the ATP, in fact, I think you could say 7/8 of the ATP. If you use 32 ATPs created for one glucose, and then only 4 are created outside of the electron transport chain. So 7/8 of the ATP is created during the last phase. And Josue, there's a theoretical possibility of getting to 34 or 38, but I think like 32 is probably a more realistic and a more commonly cited. And I just want to hit Brandon's question about how should you pace yourself on the FRQ writing portion of the exam. I think you just have to think that there's 6 questions. They're not all the same in terms of length. You're going to have 90 minutes. So the thing is just pay attention to time. And here's really the key question. If you go into the AP Bio exam having written many FRQs, you're probably not going to be surprised. So again, what I'm saying to you on this 2-week mark before the AP Bio exam is practice. Write lots of FRQs from the College Board's website. Write lots of FRQs from learn-biology.com. And if you do that, you don't have to worry about pacing because it'll be straightforward. Oh, here's the challenge. I'm ready for it. Don't set yourself up to not be familiar with the task. And the task is writing lots of FRQs. And Nahal is asking about the concentration part of the of electron transport chain. Yes, that's super important. I'm going to take you there right now. Okay, so we've got these 4 phases of cellular respiration: glycolysis, the link reaction, Krebs cycle, and the electron transport chain. I've been talking about how most of the energy is created here. 7/8 of the energy is created in the last phase. And you got to know that cellular respiration is a redox reaction. That's from chemistry. And basically what that means is that glucose gets oxidized to carbon dioxide. It loses electrons. Think about the fact that glucose is a fuel. Carbon dioxide is something that we use to put out fires. You can't burn carbon dioxide. You can burn glucose. Oxygen gets reduced. What does that mean? It receives hydrogens and electrons. Oxidation is a loss of electrons, and reduction is a gain of electrons. So cellular respiration is a redox reaction. It's an oxidation reduction reaction, and you have to understand that. Now you do. As glucose is oxidized, there are these intermediate redox reactions that reduce mobile electron carriers. Those mobile electron carriers are NAD plus and FAD. And watch how this works. NAD plus is reduced. It receives electrons from food like glucose, and that makes it into NADH. And then NADH is oxidized, and its oxidation powers the electron transport chain. FADH2, same thing. It receives electrons from food. That makes FAD into, that makes FAD into FADH2, and then FADH2 gets oxidized, and that again provides electrons for powering the electron transport chain. So those are 2 background concepts that you have to hold on to. Let's go through the whole thing right now. Here's the whole thing. The big picture is that glucose and its byproducts. What do I mean by byproducts? Pyruvate or pyruvic acid, acetyl-CoA. Those are oxidized as NAD plus and FAD are reduced to NADH and FADH2. And again, going back to Mohammed's question, The Krebs has like 10 enzymes. You don't need to know about the enzymes. You don't need to know about the intermediates. You really just need to know about acetyl-CoA, one other compound, CO2, NADH and FADH2, and ATP. Glycolysis, it's also like 10 reactions, but you don't need to know the inside. You just need to know the big picture. And I love what Ray Lowell is saying. Yeah, we're all going to get a 5 on the AP Bio exam. So in the last phase, oxidation of NADH and FADH2, that powers ATP creation, and I'm going to show you how. Okay, so there's glycolysis. Energy in glucose generates ATP and NADPH. The end product is this 3-carbon molecule, pyruvic acid. The link reaction brings pyruvic acid into the mitochondria. Remember, the mitochondria generate most of the ATP, and they convert it into this molecule, acetyl-CoA. That generates NADH. You see it right over here, and it releases 1 CO2. 1/3 of the CO2 that you exhale in every breath comes from the link reaction, right over here. The Krebs cycle oxidizes acetyl-CoA. Oxidation, taking away electrons, and it produces 3 NADH, 1 ATP, 1 FADH2, and that releases 2 carbon dioxides. So that's the other 2/3 of the carbon dioxide that you exhale. And then finally, the electron transport chain. NADH and FADH2 are oxidized to create electron flow to power phosphorylation of ATP, ADP, and let me say that again, to power phosphorylation of ADP to ATP. When you add a phosphate to something, you phosphorylate it. So that's what's going on there. Where does all of this happen? This is important to know. Glycolysis happens in the cytoplasm. The link reaction occurs as pyruvate crosses the mitochondrial membrane, enters the mitochondrial matrix. The mitochondrion is essentially a cell that lives inside of our cells, and it has its own cytoplasm, and that's called the mitochondrial matrix. And then the Krebs cycle occurs inside the mitochondrial matrix, and the electron transport chain and oxidative phosphorylation, that occurs along the inner mitochondrial membrane and the intermembrane space. And somebody asked before about gradients, and that is where you have to know it, and we're going to go there. So, glycolysis, a quick run through, but before we go there, just want to do a little survey of the chat. And things are looking pretty good. Nahal, we're going to get to your question about the inner and outer mitochondrial membrane. And somebody responded to Ryan's question. You don't need to know that much about biogeochemical cycles. That's going to come back into AP Bio next year, but you're not going to really care about that because you're not going to be taking AP Bio next year. So I teach a lot about the carbon cycle. It's so important. I have a song about it, and I have a tutorial about it. You might want to familiarize yourself with it because it's really connected to photosynthesis, but otherwise, don't worry that much about those biogeochemical cycles. Okay, glycolysis. It's got 3 phases: investment, cleavage, energy harvest. In the investment phase, there are enzymes. All of these arrows represent enzymes, and what they do is they phosphorylate glucose. What does that mean? When you add a phosphate onto something, you phosphorylate it. So this costs the cell 2 ATPs. That's why it's called an investment phase. So over here, between here and here, that's investment. Now again, big picture. You don't even really need to know the name of these 2 compounds. You just need to know that glucose gets phosphorylated with 2 phosphates, and that's it. Now in the next phase, called cleavage, this intermediate compound, I'm giving it a name because it has one, it's called fructose 1,6-bisphosphate. It's split apart into two G3Ps. That's called glyceraldehyde 3-phosphate. I would know that. That's an important term. It shows up also in photosynthesis. So know that G3P is one of the intermediate products of photosynthesis, and it's a 3-carbon molecule. And then finally, in the harvest phase, in parallel, it happens twice, once for each G3P. That G3P is oxidized as NAD plus is reduced to NADH. So here it is, NAD plus, it's reduced to NADH. It's happening down here, over here. And then enzymes phosphorylate 2 ATPs. I'm sorry, I said that wrong. Enzymes phosphorylate 2 ADPs, and they create 2 ATPs. So at the end of the process, what do you have? You have to think about this in terms of gross and net yield. You get gross 4 ATPs, 2 NADHs, and 2 pyruvates. But you had to spend 2 ATPs in the investment phase. So your net yield, which you should know, 2 ATPs, 2 NADHs, 2 pyruvates. That's what you need to know about glycolysis. I got a song about that that explains it all. You can find that. And and Ava just asked me about that. And somebody also asked, how do you sign up on your website without a code for your teacher? Well, if your teacher isn't using learn-biology.com, then what you got to do is this. You got to go to learn-biology.com/apbiology, and you'll go to a sign up area, and you'll pay for a subscription that is a monthly subscription, and you can pay, and then you can cancel the subscription right away because you're not going to want to use this after the AP Bio exam, and we don't want you to have to email us and beg for a refund either. So just sign up for a 1-month subscription. Okay, so answered that question. And Brandon, you can't find this slideshow that I'm using. This is like my core intellectual property. I use my slideshows and I make them into videos and stuff like that. So I don't share my slideshows, but I do make videos. And again, if you want an easy way to access this material, I got something great for you. And what I'm going to do is this. I'm going to stop. I'm going to present something else. Okay, I'm going to stop sharing for just a minute. Okay. And what I'm going to do is I'm going to go here. I'm going to go to my website. And I'm going to go to, I'll just go share that. Okay, so let's go like this. And share the screen, and here we go, share. Okay, so everybody, this is learn-biology.com. It's a little big. There we go. Okay, so that's a much more reasonable size. And in terms of like questions like Brandon's, so I have a course that I made for you, and that course is the AP Bio Video Pathway. So go to the AP Bio Video Pathway, and then if you wanted to review glycolysis, you'd go there. And then you can watch my video about glycolysis. You can hear my song about glycolysis, excuse me. I've got a summary with diagrams and, you know, it's a summary. And then I've got flashcards, and then I've got quizzes. So I'm not sharing my slideshows, but this is what you get on learn-biology.com, and I sure hope you use it. So let's go back to where we were, and I got to go like this. And there I am again, and now I'm going to go like this. Now I'm going to go like this, and there we are. Okay. So glycolysis rap, you can sing glycolysis. Come on, sugar for the breakdown, And literally that song was written with AP Bio in mind. It tells you exactly what you need to know about it. And everybody is correctly listing what the price is for the website. Good job, guys. Okay, so want to learn more, go to learn-biology.com. Let's go on to the link reaction. Link reaction. Here it is. In step 1, we've got pyruvic acid over here. Now watch this, because this is cool. B represents the mitochondrial membranes, both the inner membrane and the outer membrane. And pyruvate goes across a channel. I think this is facilitated diffusion, but there is one way that it gets through that costs a little bit of energy. And that's why the exact amount of ATP that's made is not clear. It's not like 30 ATPs per glucose. It depends on how that pyruvic acid is making its way across. But as it does, an enzyme over here snips off this functional group. This functional group is a carboxyl group. Is that important to know? It's useful to know, but just note that it's a carbon with 2 oxygens. If you break that off, that's CO2. So that's the first thing that happens. The next thing that happens is other enzymes oxidize the resulting 2-carbon molecule. NAD plus becomes NADH, and that powers the reduction of NAD plus to NADH, which is what I just said. At the end, there's another enzyme that attaches the 2-carbon leftover to this carrier molecule called coenzyme A, and that generates acetyl-CoA. That's the starting point. That's all you need to know about the link reaction. Now we're going to do the Krebs cycle. The Krebs cycle, it looks so terrible. And going back to an earlier question, how much of it do you have to know? Just the big picture. I'm going to show you exactly what you need to know. Let's do this. At the start, you've got enzymes. They're shown over here at number 1 that transfer these 2 carbons from acetyl-CoA onto this 4-carbon molecule called oxaloacetate. I've just mentioned 2 of the things that you should know by memory. Oxaloacetate, it's a 4-carbon molecule, and it picks up these 2 carbons, creating this 6-carbon molecule, citric acid. The Krebs cycle has a bunch of names. It's called the Krebs cycle after its discoverer. It's also called the citric acid cycle after this compound, and it's also called the TCA cycle, which stands for tricarboxylic acid, because citric acid has 1, 2, 3 carboxyl groups. So those are the 3 names. Now, once you've made citric acid, then we've got a whole bunch of enzymes, 2 through 8. You don't need to know any of those enzymes. You don't need to know any of those intermediate compounds. But what they do is they reduce NAD plus to NADH. That's going to be used in the last phase. And they also, at one point in here, they phosphorylate ADP and phosphate into ATP. That's called a substrate level phosphorylation, because an enzyme uses ADP as its substrate to make it into ATP. Substrate level phosphorylation. That's what we're talking about. Now, the Krebs cycle, all you need to know are its products. And the main thing is that for each acetyl-CoA that enters the cycle, 1 ATP, 3 NADHs, and 1 FADH2 are generated. 2 carbon dioxides are released as a waste product, and oxaloacetate over here, this 4-carbon compound, that is the starting and the ending compound. Okay? And I've got a song about the Krebs cycle. And, again, just want to encourage people that I've got videos about all of this stuff that's on learn-biology, it's on learn-biology.com. And that, those videos were made by me for you. So now we're at the best part. We're at the electron transport chain. And let's talk about what happens. In the electron transport chain, the end of all this beautiful biology that we've been learning about. What happens is that electrons from NADH and FADH2, shown at 1 and 4, they're oxidized by proteins within the electron transport chain. The electron transport chain is this arrow over here at number 2. The electron transport chain is a series of membrane-embedded proteins in the mitochondrial inner membrane. And there was recently an FRQ about that. It was the question in 2024 was like, describe the functions of the inner mitochondrial membrane. And you'd have to say the inner mitochondrial membrane has the enzymes for the electron transport chain, and it also has this enzyme, ATP synthase. So that's what the inner mitochondrial membrane does and is, and it's incredibly important. Now, some of those electron transport chain proteins are proton pumps that pump protons from the matrix to the intermembrane space, creating an electrochemical gradient. Somebody asked about that earlier, and that's what we're seeing right now. So protons are pumped from here to here. Note that it's low concentration over here and high concentration over here. Lower concentration to higher concentration, which means that this is active transport. And you know it's active transport because there are pumps, and pumps need energy. So that's an important concept, that this process over here is active transport. Why do we breathe oxygen? Because oxygen over here is the final electron acceptor, pulling electrons down the electron transport chain, increasing the proton gradient between the matrix and the intermembrane space. In other words, it's disappearing protons over here, which is increasing the gradient between this area, the intermembrane space, and the intermembrane space and the matrix. So what have we done? We've trapped all these protons over here in the intermembrane space, and that creates a huge gradient, and gradients can be harvested for energy. If you live in California like I do, somebody asked me like what time it is for me. It's 6:43. You know that a lot of our energy or electricity is generated by water that's stored behind huge dams. And when the water flows through that dam, it's used to turn a turbine to generate electricity. Cells do something very similar. There's all of these protons in the intermembrane space. There are very few in the matrix. The protons will diffuse through any channel that they can find, and there's only one. That one channel is ATP synthase. ATP synthase is a channel, and it's an ATP synthesizing enzyme. It's the only way for protons to leave the intermembrane space. And as that process happens, you need to know that that is facilitated diffusion. Facilitated diffusion through the ATP synthase channel back to the matrix powers formation of ATP from ADP and phosphate. Holy cow! We've now explained all of the ATP that's generated through cellular respiration. And I want you to know that this process of, it's called chemiosmosis, protons diffusing through, generating ATP. It's found in every domain of life. It's found in bacteria, it's found in the archaea, and it's found in us, the eukaryotes. So now let's see where we are. I've got a song about the electron transport chain, the chorus of which will tell you everything that you need to know. The chorus goes something like this. It's The mitochondrial electron transport chain uses electron energy for pumping protons from the mitochondrial matrix to the intermembrane space, increasing proton concentration in that place. The only way the protons can escape is through a channel and an enzyme ATP synthase, which uses diffusion protons kinetic energy to make ATP from ATP and P. Just that chorus basically tells you almost everything you need to know about the electron transport chain. And I hope you enjoyed that little song right there. So there we are. Tropical Vibes asks for an explanation of this. So this phylogenetic tree, this is like the big phylogenetic tree of life. And basically what it's telling you is that from the last universal common ancestor, sort of the first population of living things, life split off into three directions. Those three directions are domains. One is bacteria, the other is archaea, and then our domain, the eukaryotes, which include the complex multicellular beings like you and me, but also plants and crabs and every animal on the planet. We were formed by a fusion of these two domains, and that's a quite remarkable process. So I hope that does the trick. So that's the electron transport chain.
Now all these protons in the intermembrane space,
listen to this on your own. I don't want to be
out of that place.
Nice about your time. So there was a request to talk about thermogenesis, but before we do this, any questions? Like, did I get everything that you needed me to get about, the electron transport chain? You guys have any questions or anything like that? I'm scanning the chat, and there is a very, very useful conversation that's going on. Austin is telling me about, well, Austin, I'm going to say something I've said before. Austin is asking me about what I can do, any tips on that stuff. And the tip is you got 2 weeks and you got to practice. And you got to practice from two sources: the College Board's FRQs and my FRQs, because I have FRQs with AI feedback on learn-biology.com that will just blow your mind. So that's what I would do. Write those a lot. I'm going to hold off on questions related to Unit 7 and 8 because we're going to do them in another one of these sessions. All right, so somebody asked about thermogenesis. Let's talk about thermogenesis because it's totally cool. So the idea about thermogenesis is that cellular respiration can be used to generate heat instead of ATP. How? Well, first of all, who does this? The organisms that do this include newborn babies, like newborn human babies, and hibernating mammals, like bears. They have brown fat cells. Now, you guys who are watching, society might treat you like children, but effectively, biologically, you're adults. And you don't have that much brown fat anymore. But you did when you were a little baby. And the brown fat cells are dense with mitochondria. So when body heat is needed, there's a hormone that induces a proton channel. That's not a misspelling. It's a channel for protons that's called thermogenin, or UCP, which stands for the uncoupling channel. And it forms in the inner mitochondrial membrane. So here's the whole electron transport chain. Here's ATP synthase, and hormones will cause this additional channel to form. Protons can therefore diffuse through thermogenin, or the uncoupling channel, and as they do that, they bypass ATP synthase. Now, why? Because electron flow along the electron transport chain, it's referred to as the respiratory chain over here, but it's the same thing, that generates heat. Just like when you turn on the toaster and electricity flows through those wires and they turn red, well, that happens to a smaller extent in your cells every time you're running the electron transport chain, and you're running the electron transport chain all the time. Now, the bear that's hibernating doesn't need ATP. It's hibernating. It doesn't need to be contracting its muscles and stuff like that. And in fact, if there were ATP that were to build up in the cells, that would be harmful. So this is a way of generating heat without generating ATP. So that is the basic idea. We're about to talk about anaerobic respiration and fermentation. I know that there was a question about that too. But I do want to emphasize that we've got on learn-biology.com so many tutorials about all this stuff. And again, I want to encourage you to go ahead and sign up for learn-biology.com. You'll get access to FRQs, you'll get access to multiple choice questions, and might as well tell you about this. In addition to these YouTube lives, I'm also running a series of smaller reviews that are paid events that I'm doing on Zoom. And if you go to the URL on the screen, apbiosuccess.com/exam review, you can learn about those and you can sign up. And again, those aren't free. They're on Zoom. And you know, Zoom, it's much easier to ask questions. It's much easier to get personal attention. So just know that that is available too. And Montserrat is asking about signal transduction and feedback loops. I'm going to do that, but I'm going to do that in another one of these lives because I think we're going to be pretty, let's see where this goes, but I think we're going to be pretty full up just talking about Unit 3. We haven't even talked about photosynthesis yet. Let's talk about anaerobic respiration and fermentation. Here's the big picture. For aerobic respiration, you need, let me put this down. For aerobic respiration, you need oxygen. It involves the whole shebang of what we just talked about. Glycolysis, the link reaction, the Krebs cycle, the electron transport chain, generating about 32 ATPs. Most of the ATP is created in the mitochondria. In anaerobic respiration, when does it happen? Well, it's when oxygen is lacking or insufficient. If you're sprinting, if you're doing push-ups, in all of those anaerobic sports, if you're lifting weights, you can't get enough oxygen to your muscles to continue the muscle contraction. So eventually your muscles will switch over to anaerobic respiration. It just involves glycolysis followed by fermentation, and it just generates 2 ATPs. Can you survive on that? Not for long, but if you need to generate those ATPs so that you can fight or flee, anaerobic respiration is a fantastic thing to be able to do. It occurs solely in the cytoplasm. All right, so that's the story on the big picture level about anaerobic respiration. Fermentation is a part of anaerobic respiration. Fermentation is glycolysis followed by reactions that regenerate NAD+. Why? Because you can't do glycolysis without NAD+. In other words, to create these 2 ATPs, you need to have NAD+ around because it's a substrate for some of the reactions of glycolysis. And 2 ATP are better than none. So that's why your cells do anaerobic respiration. They do it through fermentation. You need to know about 2 forms of fermentation. The first is ethanol fermentation. Now, we don't do ethanol fermentation. Yeast do ethanol fermentation. I have a little bottle of beer with the foam over here. There's some yeast, and yeast, what they do is they do glycolysis. They result in pyruvic acid, and then there are reactions that remove a CO2 from pyruvate. They chop this piece off over here. That CO2 makes up the bubbles in beer. That CO2 makes up the bubbles in bread. That's why bread rises, because yeast are doing this. And then there are other enzymes that take this compound, acetaldehyde, and they reduce it to ethanol. Reduction is gaining electrons. So they gain electrons, and that's why alcohol is a very calorie-rich product. Like, this idea of a beer belly, you can gain a lot of weight from drinking alcohol because it's a very energy-rich food. Why is it energy-rich? Because it's a reduced product of acetaldehyde, which is made from pyruvic acid. Note that as this is being reduced, it's gaining hydrogens. NADH is losing hydrogens, and it's becoming NAD+. And then, fantastic. You know what we can do? We can do more anaerobic respiration. We can do more glycolysis. So that is what's going on there. Lactic acid fermentation is what you and I do when we do anaerobic respiration. Every time it occurs in muscle tissue under anaerobic conditions, and I have a picture of someone doing push-ups. I'm not gonna hit the ground and do them now, but I can do almost 50 push-ups because I've been working on that. And that's an anaerobic activity for me. What happens there is simpler than alcohol fermentation. We take pyruvate, and in one step, we reduce it to lactic acid. And as we do that, NADH is oxidized to NAD+. Why? So that glycolysis can continue. All right, let me look at the comment stream. Okay. Doing a little scanning over here. Things are looking good. All right. I'm not much of a weightlifter in response to some of these questions. Now, let me just poll the group over here. So I've got about a half an hour left. I was planning on doing this for about 90 minutes. We could do some multiple choice questions, or what might be better is I can walk you through photosynthesis. So why don't you put in the chat, let's do photosynthesis. Let me get a sense of the people here and what you want me to do in the half hour that we have remaining. So I'm going to take a sip of tea from my Science Music Videos cup, which you can buy on the Science Music Videos channel. All right, I'm seeing two people talk about multiple choice questions. No one's voting for photosynthesis. It looks like it's an almost unanimous choice. Okay, now let me just tell you the complicated thing about multiple choice questions. I'm going to stop doing that. I'm going to go like that. I'm going to go like that. Here I am again. And now I'm going to present this over here. Okay, so let me present this and share my screen and multiple choice practice. Okay. All right, so let's go like this. Okay, so it's harder to get public multiple choice questions from the College Board. The College Board knows that its free response questions are going to be insecure once they're, used on an AP Bio exam, but they hold on pretty tight to their multiple choice questions. So there's only a few sources of those multiple choice questions. One of them is in the course and exam description, and that's where I got these. And if you want to, it might be easier for you to actually download the, go to the course and exam description, and then it's on about page 200, and it's complicated because it's a PDF. So like the number in the lower right side is going to say 200, but if you use the PDF features, it's page 207. But here's the URL. How can I give this to you? I can give this to you by going like this. Going like this. like this. Okay, beautiful. Yeah. Okay, so I just put the address, the web address, in the chat, and you can download it and go to page 207, and that might be an easier experience than you just doing this entire thing online. But let's do it. Okay. So here's the deal. When I say, I want you to think about the answer. Don't put the answer in until I say send it. Okay? So otherwise you're going to ruin the experience for everybody else. So here's, I'll read the question aloud. Insulin is a protein hormone that is secreted in response to elevated blood glucose levels. When insulin binds to its receptors on liver cells, the activated receptors stimulate phosphorylation cascades that cause the translocation of glucose transporters to the plasma membrane. Based on the information provided, which of the following best describes the role of insulin in this liver cell signal transduction pathway? And I think right now I'm having a great idea that I think I can do this. Okay. Yeah. Let's do this. I'm setting up my screen so that I can share the entire screen. And I think that is going to be fantastic. So, guys, you can't see this yet. I'm going to stop the screen for a second. Now what I'm going to do is present again. I'm going to share the screen, and I'm going to go to the entire screen, and there we are, and fantastic. Okay, so the only problem is that it's possible that this might be a little small, but I see people are already putting in the answer, and the answer is indeed A, and I want to show you something. If you go to learn-biology.com, we're going to go to the AP Biology curriculum, the one that your teacher would be using. We're going to go to cell communication, and we're going to go to signal transduction. Okay, and you see that diagram over there? Okay, let's see who's really got this stuff. Give me the number of the receptor. Give me the number of the receptor in this diagram on the right. Send it right now. Send the number of the receptor. Where's the receptor over here? Show it to me. Give me the number. Yeah, the receptor is 2. So in this diagram, what could be insulin? What could represent insulin? Send the number. Send the number. What could represent insulin? Yeah, insulin is the ligand over here. And I wonder if I can get a better pointer. Can't really. It's too bad. Okay, so you'll have to just see my cursor over here. It's very small. But this would represent the ligand, which is insulin. It's going to interact with the receptor. And then what we're going to have is, you might remember from Unit 4, somebody asked me about it, and I said no, but I guess I'm doing it now. What we do is we activate the G protein. The G protein activates a membrane-embedded enzyme that's called adenylyl cyclase. And adenylyl cyclase converts ATP into cyclic AMP. That's the second messenger that sets off the signal transduction pathway. So insulin acts as a ligand. Fantastic. All right, guys. Answer is A. Another question. Don't send the answer until I say send it. Humans have a diploid number 2n of 46. Which of the following statements best predicts the consequence if meiosis did not occur during gametogenesis? I don't know why they have to use words like gametogenesis. It's just like gamete formation. So look at the choices. The gametes would get larger from one generation to the next. B, the chromosome number would double with each generation. C, the chromosome number would be halved with each generation. D, the chromosome number would triple with each generation. I'm going to give you 10 seconds to think about it. Then I'm going to say send it, but don't send it yet. Send it. Okay. If meiosis did not occur, you got to read this super carefully. If it did not occur, let's look at a big picture of meiosis. Okay, this is a good big picture of meiosis. Beautiful. Let me make that bigger. Okay, what does meiosis do? Meiosis is reduction division. It cuts the chromosome number in half. Notice that we have notice that we have 4 chromosomes, 2 homologous pairs over here. The first part of meiosis is a doubling of chromosome number. So now we have 4 chromosomes, but they're each doubled, and they've got 2 sister chromatids. In meiosis 1, homologous pairs are pulled apart. In meiosis 2, sister chromatids are pulled apart. Now, there's so much more that happens, but not really relevant to this question. But that's the deal with this question, and that is why the answer was, B. The chromosome number would double with each generation if meiosis did not occur. Okay, so everybody just got to stay loose, and that's how you get these questions right. That might have been the least useful information that I gave you. Okay, folks, here's some crazy sounding acronyms. Mutations in MYO6 and POU4F3 genes have been associated with a form of hereditary hearing loss in humans. Researchers studying the genes have proposed that POU4F3 encodes a transcription factor that influences the regulation of MYO6. Which of the following questions will best help guide the researchers towards a direct test of their proposal? Choice A. Have mutations in other genes also been associated with hearing loss? In what type of cells are the mutant forms of the POU4F3 gene expressed? Are mutations in the MYO6 and POU4F3 genes also found in mice? Do mutations in the POU4F3 gene affect MYO6 mRNA levels in cells? I want you to think about this, and I want you to put in your answer, but guys, don't send it until I say send it. So you got 10 seconds. 9, 8, 7, 6, 5, 4, 3, 2, 1. Send it. Let me see all those answers. Come on through. Okay, the answer is, I'm having a little trouble with double clicking. The answer is D. Okay, so they are researching hearing loss. So they're not really going to look for mutations in other genes. So we can eliminate A. And a big part of multiple choice questions, it's just like getting rid of the stupid ones. In what types of cells are the mutant forms of the POUF3 gene expressed? Well, we know they're expressed in these cells that are associated with hearing. So in the same way, that's not going to let them test this. And what are they testing? Researchers have proposed that POU4F3 encodes a transcription factor that influences the regulation of MYO6. So that's not going to help. Are mutations in MYO6 and POU4 genes also found in mice? That's not going to help. So I just want to say, like, you could use a process of elimination and get to D. But let's think about this. If POUF3 encodes a transcription factor, what's a transcription factor? I'm going to find a diagram for you on learn-biology.com. Let's go to Unit 6. Let's go to eukaryotic gene regulation through control of transcription. Okay, so this is a brutally complex diagram over here. But the main thing that I want you to know is that transcription factors are like this whole mess over here at G and H. And what they do is they let RNA polymerase bind. Why? So that transcription can happen. And what does transcription create? mRNA. And that's why the answer to this is D. Do mutations in POU4F3 affect this mRNA levels? That's exactly what a transcription factor would do. Okay, so let's do another question. All right, so I broke this question up into several parts, and let's read it aloud. In the early 1970s, researchers hypothesized that carbon was the limiting nutrient in many aquatic ecosystems. Sorry, throat's getting a little sore here. To test this hypothesis, the researchers divided a small lake in two roughly equal halves with an impermeable curtain that was fastened and sealed to the bedrock of the lake. Beginning in 1971, the researchers treated one side of the lake with sucrose and the other side of the lake with both sucrose and phosphate. From 1971 to 1983, the researchers monitored the phytoplankton biomass in both parts of the lake. The results are shown in Figure 1. So I hope that's big enough for folks to see. I'm going to make it a little bit bigger. Let's see if that'll work. It's not doing anything. All right, well, sorry. Uh-oh, what happened there? That wasn't what I wanted to do. Okay, got to bring that up again. Sorry. This was practice MCQs. Okay. Oh, we had a little bit of a delay here. Let's go here. Okay, good. It's pretty normal looking now. So I can't make this really bigger. So just sort of take a look at this. This dark line with the black dots, that's treated with sucrose and phosphate. This bottom line is just treated with sucrose. So just sucrose, sucrose and phosphate, that's really all you need to know. So here's the first question. Which of the following claims is best supported by the data? Carbon was a limiting factor for phytoplankton. Phosphate was a limiting factor for phytoplankton. Both carbon and phosphate were limiting factors. Neither carbon nor phosphate was a limiting factor in the lake. Folks, think about that for 10 seconds. Send me your answer, A, B, C, or D. Go, do it. I put the graph really small in the top left. And again, the reason why this is tough is because it's hard to see this, and that's why I encouraged you to, I don't think I still have this in the chat, do I? Oh, yeah. So if you go and grab the course and exam description and just go to like page 207, you'll start to see these questions. So here's the issue, and I'm seeing the answers come in, and the answer is B, phosphate was the limiting factor. Let me go back to where this is a little bit bigger. Okay, this is about as big as I can make it. So this is sucrose plus phosphate. This is just sucrose. They both have sucrose. One is much, much higher than the other. And that means that it must be the phosphate that's making the difference. Hence, the answer to this question is B. Phosphate was a limiting factor. Now, what's a limiting factor? So let's go here, and let's go to here. Let's go to Unit 8, Ecology. And let's go to Population Ecology over here. Okay, limiting factors are what cause population growth to level off. So a population will grow exponentially, and then it'll be limited by some factor in the environment. Let's see if I can make that bigger. And where do I have it? Yeah. So limiting factors do exactly what they say. They limit population growth. So what's the thing that's limiting growth here? It's got to be the phosphate. Okay, the average growth rate of phytoplankton from 1971 to 1975 is closest to which of the following? So this is interesting because this is very FRQ-like, but it's happening at but it's happening within a multiple choice question. So just remember what they're asking you to do. What's the rate from 1970 to 1975? And I'm going to go back to where the okay. Well, I kind of ruined this question, but if you didn't know how to figure out the rate, I'm going to demonstrate it for you. They're asking you what the rate is between 1971 and 1975. So this is 1970, this is 1971, and I'm noticing that that is 1,000 because this is 2,000, this is 4,000. So this is 1,000. In 1975, the rate is 7,000. So 7,000 minus 1,000 is 6,000. We can all do that in our heads, right? 7,000 divided by 7,000 minus 1,000 is 6,000. And it's from 71 to 75. That's 4 years. So 6 divided by 4 is 1.5. So now we're just going to add a couple of zeros. It's 1,500 milligrams per cubic meter per year. All right, so that's this. And you know what? This is a fantastic question because now we've reviewed how you calculate the rate. And the rate is, again, it's rise over run, rise from here to here over the run of time. And so that's how you figure that out. And I'm just looking at the chat, and Josue, you are right. Phosphorus is the thing that's capping the growth. That's why it's the limiting factor. That's good. All right, so let's do another question, part of this series. All right, so now this is a really good question about methodology because it's asking you which of the following treatments would have been the best control treatment for the experiment. In other words, they kind of had, I'm not going to tell you more, but here are your choices. An untreated section of the lake, a section of the lake that was treated with phosphate but not sucrose, a different lake that was treated with sucrose and phosphate, a small pool of the lake water maintained in a controlled laboratory. So think about it. And when I get down to 1, you're going to hit send. 5, 4, 3, 2, 1, send it. We got the YouTube delay, and it's coming through. Okay, so most people are getting this. It's A, an untreated section of the lake. They did something weird, which is that they put sucrose in both sections of the lake, and then they put phosphate in one of those two. But that means sucrose itself is a variable, and a better control would be one that had nothing at all. That would be the best control. Okay, this is another great question. So which of the following was most likely a direct consequence of the addition of phosphate to the lake? Listen to these choices. Look at these choices carefully. The amount of biomass in the first trophic level decreased. B, the amount of biomass in the second trophic level decreased. C, the amount of energy available to producers in the lake increased. And D, the amount of energy available to consumers in the lake increased. So think about this for 10 seconds. And in 5, When I get to 1, send it. 4, 3, 2, 1. Send it, guys. And I don't know if I'm seeing the answers yet. Okay. Oh, I just have to scroll down. Okay. This is a really tricky question. Okay. The amount of biomass in the first trophic level decreased? No. I mean, here's the phytoplankton, and it went up. So there wouldn't be any decrease. The amount of biomass in the second trophic level decreased. We don't have any information about that. So sometimes you eliminate these questions based on no evidence. Now in C, the amount of energy available to producers in the lake increased. So this is kind of a tricky question. Here's a very simple ecosystem diagram showing trophic levels. So we've got producers, primary consumers, and carnivores. And just so that you know what this is all about, this is a scenario about like life on an intergalactic spaceship that I use to teach about trophic levels on learn-biology.com. Totally encourage you to do it. It's really fun. But the idea is that the amount of energy that's going to the producers stays the same. Because what is the energy that's going to the producers? It's the sun. That's not changed. They didn't do anything to shade one part of the lake or anything like that. So the energy that goes to the producers stays exactly the same. But because of this phosphate, we have this surge in the amount of producers. Why? Because we've eliminated a limiting factor, which means that the amount of energy available to consumers in the lake increase. So the answer is D. Okay. So, and I don't know if people are talking about this, but listen, C is wrong. C is not right. I'm sorry, I went forward one question. You can't see it now. The amount of energy available to producers is the same. What we did was we removed a limiting factor, and the removal of that limiting factor caused there to be more producers. That made energy available to the consumers. So that's why D is really the only reasonable answer here. And okay, all right. So folks, let's do another question. And the enzyme trypsin aids in protein digestion in the small intestine. The relative activity of trypsin, and I really hope people can see this. I think people are, because people are answering the question. The enzyme trypsin aids in protein digestion in the small intestine. The relative activity of trypsin at different pH values is shown in figure 1. So here we are. I hope people can see that. It's a little bit small on my screen. All right. So which of the following statements best explains the activity levels of trypsin shown in figure 1? A, the small intestine releases inhibitor molecules that block the activity of trypsin until it is at its optimum pH. B, the number of effective collisions between trypsin and its substrate increase at higher pH levels. C, as pH values increase, the substrate concentration decreases, leading to an eventual decline in the rate of trypsin catalyzed reaction. D, at extremely low pH values, trypsin is denatured and cannot function efficiently. Folks, think about that question. And when I get to 1, I'd like you to answer that question. 5, 4, 3, 2, 1. Send me the answer. Okay, most people are getting this. Pretty cool. Want to show you something. So here's learn-biology.com. I'm going to make it a little bit bigger. Over here, make it, drag it over this way, like that. Okay, fantastic. So here's the teacher tutorial. Like if you were using this in your class, your teacher would be using this, and they could do something like this, which you can't do, but I can do it, which is this. They can get like all of the questions to just sort of cascade down. So this is the typical curve of enzyme interaction. And just to jump right to the question, here's a very straightforward example of how an enzyme is affected by its environment. An enzyme is placed in solutions with pH 1, pH 2, pH 3. As shown in the graph, which pH level shows the highest level of activity? Well, it would be number 2, right? And I just want you to know what you get with learn-biology.com. You get this kind of interactivity. So in terms of trypsin, it gets denatured over here, but it's functional at those high pH levels that you'd experience in the intestine. So this is actually a very similar thing. And note that this is very different from how temperature and enzymes work. And enzymes will increase their activity with increasing temperature up to a certain optimal point. That point tends to be about what our body temperature is, 37 degrees C or 98.6 Fahrenheit. But then what happens? The enzyme starts to denature because of the heat, and that makes the enzyme ineffective. Folks, I want you to know something that is very important to me, which is that I really want you to have this great experience studying for the AP Bio exam. And so I really want to encourage you to do this. Not that. This. I want you to download the plan. Go to apbiosuccess.com/plan. I want you to write lots of FRQs. We did MCQ practice. I promise you I'm going to do multiple choice practice in another session. I said that backwards because I'm getting tired. I want you to write lots of FRQs. We did multiple choice practice tonight, but we'll do free response practice in another session. And I really want you to check out learn-biology.com, our enhanced practice FRQs, because it's like somebody's going to be sitting by your side as you write those FRQs. You'll get fabulous, fabulous feedback. Now, somewhere I put on my website, let me see if I can bring this up. Hold on a sec. Okay, let's stop presenting. Because somebody just asked me a really important question. And that question is this. Okay, here's what you're going to do. So I want you to do this. I just put it in the chat. And I'll bring it up. Fantastic. So now I'm going to present that screen again. Share screen, share screen. Okay, so you can see over here, this is my schedule of live reviews coming up. So, so tomorrow's session is one of those Zoom review sessions. Those are paid, but it's worth it. Then Thursday night, I'll be on YouTube again but I can't start until I'll start at 6 p.m. And then Sunday, you could spend 3 hours with me at one of these paid review sessions. Monday, I'll be back here on YouTube. Tuesday night, another like Zoom review session. Wednesday night, I'm back on YouTube. Saturday, that's another private session. And I don't know what I'm going to do on Sunday. Sunday, it's kind of like is a sort of desperate moment. And I don't like teaching so much on Sunday before the exam because people are like, I didn't study at all, and it depresses me. But I want to say something, and I'm going to turn off the screen. I'm just going to be me. You've got a ton of time. You've got a ton of time to learn this material, but you got to be a grown up. You have to be mature. You have to start setting aside like 2 hours a day, every single day between now and the AP Bio exam. And if you can't do that, I can't promise you anything. But if you do that and you follow my plan, you've got a really good shot. And I want to emphasize the plan depends on using my website, learn-biology.com. So folks, I'm going to sign off, but what I hope I see is 70 signups of folks who go to learn-biology.com because it is going to be your ticket to success. And I also really hope that I see you at these Zoom reviews, and I'll see you on YouTube as well. Folks, this has been a wonderful experience. Everybody just take it easy. It's been great having this experience with you, and I'm so happy. And if anybody's having like trouble, Eliana is saying something like, I tried to sign up, but it wasn't allowing me to. Here's what I want you to do. I'm going to share again. So here, I'm going to present my other screen, share screen, and go like this, go like this, go like this. See this contact link over here? All right, so just email me. Or we've got like support@learn-biology.com. Don't forget the dash. So make this a little bit bigger. All right. Support@learn-biology.com, and we'll fix whatever problems. So folks, you guys were the best. I am so grateful to you for being with me today. And I hope you go to learn-biology.com where success awaits you. Okay, guys, signing off. Everybody be well. Great being with you tonight. Hope I see you at another one of these events. Everybody take it easy. Bye.